Okay,
we can get a close look at that.
That looks the same thing.
Yeah. Yeah.
Beautiful.
Yeah, so I'm pretty sure this is a glass sponge in the family Euplectellidae, and it's in the genus Dictyolis. This is one of these deep vase sponges. Really, really beautiful. That's got a bit of a covering over the top osculum. The water in these sponges actually enters the sponge in the side here, and then it goes out the top. So that's kind of the way a lot of these vase sponges work. And then they've got probably some spicules sticking out the sides to protect them from some animals, and spicules on top there maybe to keep things from crawling in or possibly even crawling out.
Yeah, this one looks really interesting. This is tremendous imagery here because you're seeing that the spicule fringe, really fine spicule fringe around that rim. And yeah, at the top looks like there's a polychaete.
It's radial. No, I mean the spicules are radial.
Yeah.
There's something inside the sponge.
And also the other thing is I don't know if they're connected at the point in the center at the top. Chris, I can't tell. I know the one we collected yesterday was not. They were just, the spicules were radiating inward, but not connected.
Yeah, it might have got a little munched when it was collected, too. I'm not really sure. And you confirmed that it was a soft sponge, right, Shirley?
Yes, it was very fragile.
It was really rigid and brittle, correct?
No, correct. It was not, it collapsed.
Yeah, so in the genus Regadrella, they can also be a bit flexible. They're not completely rigid, but they're more rigid. They have some fusion of the spicule framework going on in that particular genus, whereas this one has very little. It just is, in general, quite softer.
Yeah. Thank you, Pilot.
Okay, Pilot.
We need to start moving then.
Great. That worked nicely.
Thank you, Tara. That was beautiful.
Happy to help.
Looks like we may have a buttress or something coming
get
out
on
up
our way
ahead,
again.
maybe more
Tina
vertical wall.
Molotova has joined us, so we'd really like to get a close-up of that yellow-black coral so that she can look at it and comment and tell us if it's something different. It's been so common today that we really need to consider if that's something important to collect.
Roger. We're doing that right now. Welcome, Tina. Good morning.
Soon partial.
Now, if we're finished with that move, aren't we?
Yes,
the
we
ship
have.
stop.
Yes,
Okay,
yes,
roger
ship stop
that.
today.
I think we have as well.
It's nice twofer. Go ahead in a little more.
Oh, look.
So now this one we see actually does have lots of secondary
Based on the time
branching.
left, we've still got an average
This is the
about
first
just over
time I think that we've seen that
Okay.
clearly,
achieve
and
waypoint
that would
2
suggest that
and
this is in the genus
Roger that.
Sauropathes.
A bit more. I think waypoint
So
2
we'll
is
wait to
25.
see what Tina Molotova from Russia, Shirshov Institute of Oceanography, has to say.
This is the first one, though, I think that we've seen like that. With that secondary
It's the first
branching.
one that's had a lot of the secondary branching.
Right.
Would you like to see the crinoid?
You're seeing
Said
it.
earlier when we saw the first one, this is one of the problems.
Smooth.
You know, depending on the growth stage of the coral, it's hard to know
I think we
if
need
it
to
is
start
completed
moving
its
up.
branching or not, and so you can be really fooled
I don't know
when
how
you're
high
looking at
this
these.
looks like.
Depending on
Maybe a vertical
the
wall
age
here,
of the
but
coral, and that's why
maybe we ought to
ultimately
do a move,
to
but
get
only
a
go
truly
10 or
accurate
15
identification,
instead of the 20. I
we would
don't
need to
know.
come back and
What do you
make
think?
How far in front of?
About 16, 17 meters.
Okay. Yeah, we might as well
Thank you for that
move underway.
close-up.
Let's just go 10 meters
Yeah, okay.
until I come up anyway.
Okay. 10 meters, 350.
10 meters, 350. Bridge, this is nav. Could I have another move, please? We'll have 10 meters range, 350 bearing, and 0.2 knots, please.
So, again, some really interesting geological features here.
So that sponge, a little post note on that sponge we just imaged. So Dictyolis has only 4 known species or described species in the genus, 2 from the Indian Ocean, 1 from the Atlantic, and 1 from the Southwest Pacific. The Southwest Pacific, it's Dictyolis starmeri. Because it's the only one that's ever been described from the Pacific, we kind of defaulted to that in Hawaii, the species we have there. So I think there's an excellent chance that this is going to be a new record for the Marianas. And also, Tina Molodtsova's collaborator there at the Shirshov Institute of Oceanology in Moscow, Konstantin Tabachnik, was the sponge expert who described this genus.
And as you said, we collected a sample yesterday. It wasn't complete, but we did get the top. We snipped it off a little bit, so we don't have the base, but we do have, I would say, probably about 3/4 of it. And I very carefully have the top of the sponge in a separate little glass vial. All right, so Tina, when we see another Bathypathes, we will go ahead and zoom at the base of that stalk. Okay, some more bamboo.
Still on your screen, can we get a quick zoom on this unbranched width on the right?
Yeah, Jeff, can you go ahead and get a zoom on that one? It's about 5 o'clock, please.
Surely. Surely. Okay, video. Good partial. Hold there.
Okay, pilots.
Okay, so we're getting an image of the base of this unbranched
why don't we go ahead and repeat
coral.
that move?
You can actually see the
Another 10 meters.
nodes. Oh, nice.
350.
Okay.
Copy that. 10 more meters, 350. Bridge, this is nav. Could I have the SAM again, please? Range 10 meters, bearing 350, speed 0.2 knots.
So how do they attach to the rock? Is there some kind of cement that they use to attach?
Good copy, Bridge. Thank you.
Yeah, so the way I interpret it is that the skeleton that is deposited by the tissue, that basically it's amorphous and forms this hold path at the base. You know what's really fascinating about this one are those 2 little scraggly branches down below. Wow.
Yeah, I saw that. I was wondering if that was part of.
Learn about for
yeah.
some time.
I was wondering. Well, now Yeah, I was wondering if that was part of this bamboo coral, or it was something else, but it did. And they were like, they didn't have any polyps or anything on them. We'll try.
Yeah, exactly.
Well, you know, let's see.
Little, what looked like little branches to me,
yeah,
and they weren't covered by tissue, so it's possible that the polyps would subsequently develop. Very interesting.
Take a look at this sponge.
Yeah, another taller vase sponge. The top of the screen will be coming into your view. Wait, you're on.
Okay.
Looks like the rocks in this outcrop are not as well indurated.
Okay, now this
that's
one
different.
looks different. Chris, are you around? Do you think this is another euplectellid?
Yeah, sorry, I was blasting away in the chat.
Oh, okay.
Long message. Yeah, this is a euplectellid, it's a vase sponge, and I wonder if we could look at the bottom of it a little bit. It's dead.
We are.
That's for sure. And
well, look at the bottom of it. The bottom of it still looks like it's alive. It's white.
Like
anyway, go ahead, Chris.
Covered.
yeah, so it's probably just a larger Dictyolis. I'm looking at the horizontal rows of the oscula across the face of the vase. I was looking at the very base of the sponge because that's informative how these vases attach to the rocks. A bunch of them, including Regadrella and Dictyolis, glue themselves on the rocks with a plate. And there's another group in Corbitella that's in the genus Corbitella that actually has fibrous connections. That looked like a plate, so I'm going with Dictyolis.
Yeah, and for this one, you don't see any kind of sieve plate or anything across the top, but of course that just could be because this sponge is either dead or dying. But there was no sieve plate on the top.
Of course.
Go ahead now. A watch lead. Yes, yes. At the rate we're going now, we should be in waypoint 2, and I would say, bear with me just a second.
So you're up on top of this buttress, falls off to either side there, it looks like right now.
Okay, don't want to lose my footing then.
Yeah.
Oh.
It's a long fall.
Yeah. Like those brittle stars jumping off the coral or whatever they were on. I saw 3 of them come off. I wonder if they were disturbed by our presence somehow.
Could be.
Yeah.
They look very depressed. Now, I think we're going to need another move in here. Let's see, our heading. I think we should go for a 20-meter move this time. And
some of the other partners from NOAA who are supporting,
looks like 350 still it.
Are the NOAA Deep Sea Coral Research and Technology Program. Stand by, everyone.
Well, actually, my estimation is we've covered the same distance in about 50 minutes. So We have approximately 1 hour 40 minutes left.
Yeah, you're at the end of your leash there.
That would leave us
Sorry.
not much time along the 250-meter path to get from waypoint 2 to
Say again?
Do we have a move in progress?
dive end. It is
No,
possible.
they're discussing
but there
on
wouldn't
where
be
to go.
much,
I think
many stops.
whether to head to the waypoint end or
Okay.
go
So
continue
about
up.
50 minutes, I would estimate, to get to waypoint 2.
No, look for something.
About 50 minutes,
I think I'm pulling you back.
50 minutes.
Okay. Okay, so our navigator is telling us that we have about 50 minutes if we continue at the same pace to get up to waypoint 2, which was not up at the top of the ridge, and that wouldn't give us much time at all to
go to the
along
west.
to the west,
Yep.
because we have to come
Bridge,
off bottom in
this
about
is
an hour and a
novel.
half.
Right.
So we can pick up the
Hello,
pace.
Bridge. Could I have a 20-meter
Yeah,
move,
we have
please?
an hour
20
40 minutes left
meters,
at this point,
the bearing
but
of
we would not
350,
be able to
speed of 0.2
move
knots.
from waypoint 2 very far along the ridge. Get a little bit.
Yeah.
Or we could go up to the
We
very
have
top.
about
of the
50 minutes. 50 minutes to the top, and then another 50
Well,
minutes.
Remember that to the quote top unquote to waypoint 2
Yeah,
is
it's not really the top.
the top.
Yeah, you're
so
right, you're
if
right.
we want to continue upslope, we should think
French
about
stuff.
that pretty
It's like
soon.
a base of something there.
Okay, so that looks like it's what's left of a
Thank you,
sponge
Rich.
Okay.
spicule stalk.
Yeah, and actually that was a pretty good example of that, what's called a basiphytus attachment, that plate at the base of the stalk, and that's how they kind of glue themselves right onto the rocks. And we've sort of seen those fibrous attachments on other sponges during this cruise. So those are two important things to look for when you're trying to figure out what sponge it is.
So I was mentioning that another NOAA partner is the Deep Sea
So,
Coral Research and Technology
did they,
Program.
decide whether we're going to
NOAA
waypoint 2 or
Fisheries is
straight
another
up, or
important
that's
partner.
still up for grabs yet?
And the National Center for Environmental
I think they
Information
will-
as well. That's in addition to
Waypoint
the
2 is
Office of Marine and
required.
Aviation
It's a
Operations
decision
operating
then
the ship,
how far to go on
and the
from that,
program
whether to go
that
straight up or whether to go over
designed,
to dive end.
supported is executing this mission is ocean exploration and research.
Dive end is still possible, but it doesn't. It means we can't stop.
Roger.
And the Global Foundation for Ocean Exploration is another important NOAA partner who are operating, maintaining, upgrading, designing new things for these remotely operated vehicles.
And building them. So they have constructed most of this ROV, right? Is that true? No. I'm getting a no from
I
Shirley.
don't think so.
Excuse me.
I'm not sure. I think it started out, there was a base.
Oh, yeah. But they have built a lot of, or put together a lot of the
Oh,
pieces.
yeah. Wow, look at this.
Mm-hmm.
Okay, Don, you moving towards me, you think?
Yeah,
This
I
looks
think
like
we
the
are
edge of the
starting
Grand
to move
Canyon
now.
to me.
Here we go.
Or like partway down the slope, that is, and you're standing on a promontory looking up at the layers above you. Oh, here's another forest of things, Shirley. And what looks like from the upper left to lower right at a steep angle on the right-hand half of your screen, I am guessing we are looking at a fault trace there, as we were before, with a very slight offset. This looks like a normal fault. In other words, the upper block slid down to the right, that is eastward. Yeah, it seems to continue as we go up the slope. The offset isn't very large, but it's pretty obvious. And the rocks in the area where the faulting occurred are all sort of ground up.
Attention on the Explorer. The ship's door is now open and featuring our fire alarm special. The ship's door is open for the next 10 minutes.
What do you suppose a fire alarm special is?
I don't know. Maybe burnt caramel cookies.
We're still traveling up this fault trace right in the middle of the screen. You can see how
Nav,
the
why don't
rocks
you go
are
ahead
just
and have them
ground
tack on
up
another
into
20 to
finer
that move.
material as we move upslope.
Copy that. Bridge, this is Nav. Hello, Bridge. Could I have the same again, please? So, range 20 meters, bearing 350, and a
So,
speed of 0.2
Pathy
knots.
Pathy, Star Pathies, many, many, many of them. And Iridogorgia, beautiful, spiraling coral that was just passing over.
Thank you, Bridge.
We are coming to you live from the NOAA ship Okeanos Explorer out on the Mariana Trench at a feature called Explorer Ridge. I'm Shirley Pomponi. I'm from Harbor Branch Oceanographic Institute, Florida Atlantic University. I'm the biology science lead. Yes, go ahead, Scott.
Does that Metallogorgia on the right have a coil in it, or am I just seeing the jumping video maybe is the problem?
I don't see a
Okay,
coil.
I see what you're looking at over on the
Really?
far right. Yeah, look. Let's look on
okay.
the far
Oh,
right.
It has a kink.
I see what you're looking at.
Yep, looks like it has a kink.
Yep. Pilot, can we? Yeah, you're already heading over there, Jeff. Thanks.
Well, that's pretty odd looking.
Indeed.
Partial.
Holding partial.
What I want to do here, maybe get down lower and
It's
look up.
bowing down to us right now. Yep, sure does have a little kink in it.
How accommodating.
Oh, look at that.
I can take a bit more zoom.
Yeah, I'm going to just tilt down a little bit and fly in some more.
Sounds good.
What are you thinking, Scott?
I don't know. That's strange. I've never seen a Metallogorgia with a kink like that. I presume that has something to do with the local currents at the time that it was getting to about that size. But thanks for that close-up. We can move on, and we'll just make a note that that can happen.
Okay, great. Thank you, pilot.
Just get this. Focus. All right.
Oh, I think I'm drifting back again.
That's
Okay.
fine.
All right.
So we're at 1630 meters, 29 meters. So we got about 20 meters to go to get up slope to waypoint 2.
Catching up to me, Don.
Roger that. Yep.
That was
Not very far.
Come
and that was
back in
Patty
towards the
Fryer,
center.
the geology science lead
yeah.
from the University of Hawaii, and we have quite a
Looks like
few
we
other
probably just keep
shore-based members of our
Yeah, I was going to change the
science
heading
crew
to
for
north,
right,
though.
But I
today's
happen to be
style.
on the ship. Sitting next to Shirley.
Two of us on the
Yeah,
ship,
let's.
and we have
How far are we along on that
joining
move,
us
Richard?
from
We almost there?
their
Yes,
we are. We've got about
institutions,
another 2 or 3
offices,
meters.
or depending on what
Okay.
time it is,
Yeah, go ahead and
homes.
give us.
Due north?
Through the magic
Yeah,
of
due north
telepresence.
and-
We have a
That's me looking
number
north.
of colleagues, biologists, geologists.
Yep.
Should start flattening
We
out.
have Asako
Yeah,
Matsumoto
you
from
wanna try
the Chiba
30 then?
Institute of Technology in Japan.
It's flattening out for the
Bruce
ship for
Mundy
you guys,
from, Mundy from
by the
NOAA
next move, it
Fisheries
should be
in
flattening out.
Hawaii.
Yeah, okay.
Chris
I would
Kelly,
stick with the 20 for this one.
All right, roger
University
that.
of Hawaii. Deb Glickson, Florida Atlantic University, Harbor Branch.
Bridge,
Hong
this
Kong,
is Nav.
I think is at the University of Hawaii right now.
Yeah,
right?
she's visiting us from China.
Right,
Could I
Les
have,
Watling, also
this
University
time
of Hawaii,
range
but I think he's
20,
in Maine.
Bearing, due north, speed 0.2
Scott
knots.
France, University of Louisiana, and steamy hot Lafayette. We have Tara Harmer Luke in
Good copy, Bridge. Thank you.
stormy South Jersey from Stockton University. Tina Molitsova from the P.P. Shirshov Institute of Oceanology in Moscow. And Sonia Rowley, who's also from University of Hawaii, I think?
Not sure.
Not sure. Who have I missed? Bob Sturm may be on the phone. He's from University of Texas in Dallas. And Bob Carney may also be on the phone, or I know he's not in the chat room. Anyway, we have a very diverse,
The white stuff?
multidisciplinary, but highly expert, shore-based science crew. We're looking at this rock.
There you go.
Okay, folks, finally we do get a look at the inside of this material because this rock has broken off. It's so pale in color that it sure looks like sediment to me. And the exposed surface to the right and left is covered with a dark black manganese oxide crust. And this finally gives us a look at the interior of some of these layers. I don't see any distinct layering within this what looks like a layer in and of itself within the outcrop surface on the wall in front of us. But it's good to see into it, finally. Okay. And right above, there's a little bit more of the light color. It looks as if all of the material, starting at the upper white layer to where the little rock is broken off, that whole thing may have just sort of slumped down a little bit and exposed more recently, the material of which much of these sequences is composed.
Just above it.
Okay.
These big slabs on the right-hand side.
Walk me to it. Oh,
right there.
Well,
there.
yeah.
Is that something interesting?
We have a globular thing here.
A globular thing.
Right by the right laser.
Globular thing.
Laser points are the red dots. They're 10 centimeters apart, which is about 4 inches. And
Take
this
a bit
globular
closer
thing
zoom
we're
in.
getting closer to is about 2 inches in diameter.
It is something squishy.
Yeah, for sure. And
Let's go in.
do you think it's attached to the wall? Looks like it has some sort of attachment.
Huh. It looks like that slime urchin that we saw before, but I don't know. I don't see any central opening in it. A rock pen?
Yeah, I am at a loss.
Yeah, I don't know.
We're going to need to move
I don't
some
know. I have no idea.
80 degrees looking down.
Okay, a few more seconds.
Nobody has said this today, but just poke it.
How rude.
It's an octopus.
Full wide.
Yeah, I'm sort of leaning toward a mollusk of some kind. I don't know about an octopus or maybe a sea slug. I don't know.
We'll chalk that up to mystery of the deep.
So we may have some more of those anemones, or is that just discoloration? Yeah, it's just discoloration.
Nav, how far are we on that move now?
We have about 4 meters to go.
Okay, let's keep pushing on that same heading. 20 meters more.
Perfect. No, the ship is now stopping. So, same heading.
Same heading.
Another 20.
Yep.
Bridge, this is Nav. Could I ask for same again, please? Which is a range 20. A
So
bearing
now what's
of
all
due
that
north
white
and a
stuff
speed of
there,
20
Patty?
knots.
Getting to it.
Getting to it. Getting to it. Oh, wait for it. Wait for it.
Just trying to type in up.
A little bit of zoom there, Tara.
Actually, I think below the screen, what we were looking at is little ripple marks. What happens when the current moves across a sedimented surface? Often heavier material gets left behind, lighter material gets picked up and wafted into the groove
we are steaming
on
along
the
now.
down current side of a ripple.
Roger.
And so I think the white lines we're seeing are accumulations of foraminiferal tests that mark the down current side of these little ridges, ripple marks that are forming on the seafloor as the current sweeps across the face of the outcrop
Do a
from
quick
left
zoom
to
here,
right.
Tara, so
The
we
pilot's
can
been
see
telling
that.
us that's the
Sounds
direction of the current.
Tests.
So it probably is a current
Roger.
that is customarily
not
moving
quite
in that
full.
direction.
There you go. There's a good.
Are we
That's
zooming in on the white stuff? Because I think there might be an umbellula sea pen, which would be the first of the leg, on the to the left of the ripples that are above the ones that are zoomed in now.
Okay.
Or a second ago.
Yep, I see what you mean. Okay, hang on. Pilot, in that upper layer with the white squiggles on the seafloor, very top of the left-hand side of the screen, there's a C-pen that we want to look at.
All right.
Do you
Okay,
see
you
that?
don't have much time. We're almost over top you, and we're in a move. So,
Okay,
okay,
understood.
so-
So zoom in with me, Tara.
Sure.
There we go. I'll walk it to the middle.
Here we go, Scott.
Yeah, I can tell you what this is. This
Okay.
is what I was predicting we would see when we were on those really deep trench dives, the 4,000, 5,000 meter dives.
Okay.
This is a sea-pen. It's another octocoral, and it's anchored in the mud, so it'll have an inflatable base in the mud, and then it has that very long, thin stalk, and then at the top here you see these really giant polyps. These are some of the biggest polyps in the octocorals, long octocorals. This is the genus is called Umbellula. It's very distinctive because of that long stalk with the big polyps on top. But there are many different species, and there's a lot of confusion still on how to define the different species and exactly how many there are. So thanks for that view.
Oh, you're most welcome, and thank you for the detailed description.
By the way, I'm seeing a bit of an instant replay of that squishy thing we saw before, and it looks to me like there's some kind of a foot. It looks like it might be a gastropod.
A gastropod, really?
Mm-hmm.
Without
Yeah.
a shell?
Yeah, it's a
Yeah,
it's
I'm
a
looking at
it's
it
a
too.
mollusk.
Okay.
I'm pretty
Good to
sure.
know.
Okay, thanks, Roland.
Yay, Tina and I win the contest. So what did we win?
Oh, boy.
You win the
You get an opportunity to go to the ship store because they're having a fire special out
fire
here.
drill special.
What do you want?
No,
Oh,
you
look at
win
the coiled.
Oh, look at this coiled one.
Oh,
Okay, let's take a look at
gorgeous.
that. Take a look at that. Oh, but that's the one that's normally coiled.
Oh,
That's
yeah.
the Iridogorgia.
Okay. A quick look?
Yep.
It's really
Quick
pretty.
zoom.
Yeah, a quick zoom is good because there are several different species of Iridogorgia, and one of the ways you distinguish them is the distance between the coils and how wide the coils are, so it's instructive to get a good look. We can
good,
look
and
back at it later.
I'll
Okay,
take
great.
a bit more.
And there are
Maybe
shrimp
if you
on
can
it.
get the lasers back in so
8
they can
shrimp,
get
at least
size.
one, maybe
Uh-oh.
2. Very nice. Those lasers are 10 centimeters apart, about 4 inches apart. Great, thank you.
All right.
okay.
Great.
Need to push out.
Okay, for one.
Full line.
Okay, pilots, ship has arrived. We'll
Roger.
zoom.
Oh yeah, there is a whip over to the far
Yeah, it
right.
looks like it's flattening out here. You want to try a 30-meter move then?
Sure.
Okay.
Yeah, Scott, yes, I can confirm.
How
I'm looking at
about,
a picture of
yeah.
this right now, and
That
there are
heading
polyps
looks as
on
good
the
as any, I
axis.
guess, right at this point. 350,
There are
30
definitely
meters,
polyps on the
Richard.
Great, thank you, Shirley.
30 meters. Copy that.
We're seeing a lot more sediment
Bridge,
here and
this
very
is
thin
nav.
layers of material, some of which are breaking
Could I
up
have another move, please?
and forming
Well, this
slabs
time
on
I'll
the
have a
surface
30
just below the
meters
outcropping
at
layers. So
bearing
clearly this
350
material is
and a speed of 0.2
less
knots.
indurated, less rock-like than what we've seen deeper down.
So can we take a quick look at the one over on the right? Are you able
Good
to
copy,
stop
Bridge.
that
Thank
sponge
you.
over on the right? Because that looks like a live individual of the one we looked at earlier that was dead.
Okay, Tara. Partial. Great, hold that.
And I'd like to
Try and come in
see
a little more.
both the base and then right up at the top too. Probably
Bridge.
it's probably going to be probably going to be difficult to see much of the base, but we should be able to get up to the top. And Chris, I'm not seeing a sieve
little
plate
more
at the top
zoom.
right now, but I know you mentioned yesterday that not all the euplectellets had the sieve plate anyway. This is showing a nice spicule
I
fringe.
can see a real faint, thin one up there. So again, I'd probably go with Dictyolis
Oh
on
yeah,
this
you're
one.
right. You're
And,
right. Yep, you're right. You're right. There's a very fine one. Wow.
There it is.
Yep.
Okay.
Nice.
Okay.
Okay, great. Thank you. Thank you, pilot.
Wide.
How nice.
Mm-hmm. All right, so what's that thing that's all coiled over there on the right? Well, I mean, I know it's a coral, but let's look at it. Yeah, oh, good. Scott said he would like a close-up of the coiled whip when you get to it, please.
No, we don't have too long.
Okay.
Coming over
I
top.
would too, Scott.
Yeah, thanks. I'm trying to determine if this is yet a different bamboo
I
coral,
know.
or if this is a black coral, or if it's a chrysogorgia coral. There are lots of different types of corals that form these coils.
Seen partial.
Partial.
As we approach, I'm seeing that the polyps look relatively large, so it's almost assuredly a bamboo coral. And if you guys on the vessel there can see any dark nodes along there, that would be helpful. Just let us know.
Okay. I can't say anything right now. I don't know if you can
Time
zoom.
to
Can you
move.
zoom in any more, Tara?
Full zoom. Are we looking for nodes?
We are looking for nodes. Only the nose
We are
Node.
node looking.
We are.
Ooh, darkness. We want to see darkness. Circling the stalk.
Maybe down to maybe further down.
Wow,
maybe
hard to
further down
see.
the stalk.
Yeah,
Uh,
we're a bit
yeah,
further
try
on.
to the
saw
base.
one. This is a really
You saw one?
interesting one because of the way it's reflecting so white.
I'm going to zoom
Suggests
on the
that there's
base.
actually quite a lot of scales in the tissue.
Yep. Yep.
Yeah.
Oh, there's a
There's
node.
Very good.
That's
Yeah.
definitely a
Just
bamboo
above the
coral.
bottom.
Thank you.
Yep.
Okay.
Definitely
Okay. We're
is,
good
Scott.
with it. You're good with just the image.
Okay.
Yeah,
Wide.
thank
Okay.
you.
All right.
And up and out.
Roger.
Yeah, that's good, Shirley. We can move on. Thanks.
Thanks, Tara. Thanks, Jeff.
Very welcome.
Sure thing.
Thanks, Don. Thanks,
Better
Richard.
think. You better think, Dawn.
I just said thanks, Richard.
Okay, folks, we are at 1615, almost at waypoint 2, which is at 1609.
Now it's really flattened out.
Yep, sure has. And I think
Which is not surprising because that's
no,
what we
that's
saw
what
on
we
the
thought.
imagery.
I mean,
That's what we saw on the
we're at the
bathymetry.
top of the hill. So we're probably going to be seeing the same sort of things, same sort of surface for most of the rest of the dive if we stay up close to the top here. And there is a brittle star down there. Just went off the screen. We're seeing ripple marks. We're seeing lots of brittle stars, I think. Brittle stars.
Yep, we are.
Video searching up.
These ripple marks are not as well lineated as the ones we saw down
John,
at the
there's
bottom.
only 5 minutes left, but do you want to fly?
So, I don't know about you, but I'm kind of
Got a
voting.
fish in the water column,
Ah,
upper
well, you know what?
left.
No, we'll do it another time.
Fish in the
Take
water
2
column
minutes
upper
just
left.
to
Yes.
move around, figure out which one is which.
And we were just
Or something
surely
in the water
no,
column. Not sure
yeah,
it's a fish.
you're right. It's a fish.
Okay, so what
We're thinking
we're
of going
thinking
left.
about going left and staying closer to the rocks.
What do you guys think at home?
Okay, rollies and fish.
Well, let's get the fish description first and then talk about it. Ooh, it's very thin,
Coming at
beautiful
you really
fish.
fast.
Yeah, he is. Isn't he?
Bruce.
Well, that's the current. That's how. Hey,
He's a little scared of us.
Oh, you're killing me. I haven't seen it yet.
Yeah.
Okay, I think I see it
Something
coming
had to
into view.
create these ripples.
It's only going to be there a
It's gone
sec.
already, Bruce.
It's like the
Yep.
beach.
We scared it off.
So, do they want to head to waypoint
From
2?
the way that
Go a
this
little
is
bit
swimming,
to the east? Or?
the
Okay,
way you say
roger
it's
that.
going to dart
Copy.
away, I think this is one of these. Okay, I can see it now. It's one of these very pale Aldrovandia species. It's a different species of spiny eel than what we saw before, but in the same
Okay, we've got
genus.
100.
I'll
We've
have
got
to
1 hour
look and
12
see what the
minutes left, so we've actually
possibilities
got
are.
more
But we
time than
saw
I was
these,
expecting. We've
at
got
the beginning
250
of
meters
the
to get to dive
first
end.
leg in the Mariana.
That's well within the capability
So
of
again,
the speed
spiny
we've been
eel
doing.
in the genus Aldrovandia, but a different
you can see
species
where we are now, so
than
where
the one
we're
that was
Jeff, you
possibly
need to
after
push
we
out
saw
now,
the,
too,
the
as
dark
well.
one that we saw
I'd like us
earlier.
to go
I'll have to go
due west.
back and see if I have notes on
Don,
that.
are you still moving, or do you think the ship stopped?
I think we're stopped.
Okay.
Due west.
So we're talking about going west instead. So
Oh, okay.
I'm
Okay.
pointed west right
The
now.
next move, due west.
Yes.
Due west.
Thank
Yes.
you, Bruce.
So, folks on shore, we decided
So you heard that, pilot.
we're going
Copy.
to make a turn here and go west. This will provide us with an opportunity to stay close to
Close to
the
the
edge
edge,
of
yeah.
the wall.
Yeah.
And then we can
That way,
probably put
in
30
case we want to go take a look over the edge again, we can do
Yeah,
that.
we can do that relatively easily.
It's unlikely that there is no steep scarp to the north of us, and it's unlikely that we would see a different seafloor character
And since
if
we're
we went further up.
right, and since we're seeing so many interesting corals, many of which we may not have seen before, we'd like to continue exploring those as well. So we have a little over an hour to go.
Mm-hmm.
Okay, well, actually, we're now lined
Okay.
up west, and we're going to move west.
Great.
Okay, so we are lined up west right now. Shore-based scientists and folks at home, we are lined up west, and we're going to be proceeding west along this ridge.
Okay,
We
you
are
want to
at
get a move underway then?
1,613 meters.
To the west, 20 meters,
There's
or
another one
maybe
of
even
those
30, I guess. Huh?
coiled iridogorgias.
Beautiful.
270.
270.
These really are pretty.
Thank you, gentlemen. 30 meters, 270.
Do you want to see what those little white things are on the edge?
Bridge, this is nav. Could I have another move, please? We'll have a range
There's
of
a
30
ship on the
meters,
right.
a bearing of 270,
Oh,
and a
you're
speed
so
of
quiet.
0.2 knots.
Really?
Oh, there you are.
There.
Okay, go for it. There's one right
Okay.
above the lasers.
It's a big, red, gigantic
Good copy,
ship
Bridge,
that
thank you.
was off to the right.
Yeah, see, I can't hear you when anybody else is talking. Oh, it looks like a
Barnacles.
barnacle. Yeah,
I bet somebody cranked me down on your intercom.
yeah.
Okay. Patty, some of your favorite animals, the barnacles with their
Ooh, barnacles, aren't they
cirri
cute?
extended.
It's like that current hitting you broadside there.
Taking
Yeah,
advantage
sure
of
is.
all
Yeah.
this
Really,
stuff
really pushing against the current.
in the water current.
I think immediately to the left of this thing was a very small sponge on a stalk right there.
Stock of some kind.
That's
Actually,
on
the
the-
two
Let's hold hard to hold.
So, trying to get a handle on what is the direction of the current today here?
Okay,
Well,
video's clear.
Generally, the current has been moving from the left of the screen to the right as we were heading north, going up the wall. And so now, going westward, we should be going more or less into it.
Yeah, except it looked like those things were kind
It's,
of flagged over to the
They
left.
are sort of flagged over to the left.
They are definitely flagged over.
Yeah, it's very
Okay.
much north to
It's
south
north
right
to
now.
south now.
The current is north to south, says the pilot, which is consistent with the ripples we're seeing.
Mm-hmm. Absolutely.
So the deeper spots, I guess there's more of a north to south current, but in the shallower areas there's
No,
more of an
no.
east to west current,
No, just
or
the
am I
opposite.
getting that backwards?
Yeah, you
Just the opposite, Chris.
Yeah, backwards. So we're up over the top of the ridge now, pretty close to it anyway, and the currents are from the north to the south. While we were climbing the wall, it was from the west to the east.
Starters to the right.
Yeah, I guess I was referring to the shallow dives that we did earlier. Those were kind of east to west, if I'm not mistaken. Those 250 to 600 meter ones, and the deeper ones have seem to be more north to south. You can understand sort of the west to east just because that's the strike of the whole outcrop, isn't it? So the current's just moving along the wall down further, and then when it doesn't have that constraint up on the top, it's moving more north to south, perhaps.
Yep.
I don't know.
You got it. So it looks like the ROV gets wafted a little bit to the south in the current. So we're turning around north again, headed more back toward the wall. Well, the top of the wall.
Welcome,
And we
Josh,
still see these
Dave, and
very
Levi.
thin layers of sediment that are breaking apart and creating slabs below the edges of the layers. Is there a sponge on
It is.
the edge of that block?
Yeah,
on
it
the
is.
lower
Yes, it
left?
is.
And what is the white thing below it? We'll see.
Come on in video.
There you go. That looks
Maybe
familiar.
like a Farriad or something like that. And
Oh,
it
it's
looks
got
like
things inside.
Ship is moving.
Ship is moving. Okay. At 265 or thereabouts?
Yeah.
Good.
Nice.
Yeah,
video.
270.
So this sponge is a Farriad, Shirley. You nailed it. It's in the genus Ceria, and it is the erect form of what we think is Feria oca, but we're not positive. So Henry Reiswig refers to it as Feria near oca erecta. The Feria Oka, the classic Feria Oka, is more bushy, and then this is a elongate, taller morphology, and it's just now very questionable whether this is the same species or a different species or a subspecies. Who knows?
What was that
And it looks like there are a
Ready to
couple of
pull out
shrimp
for
in
full
there too.
specimen?
Sure.
Let me also check my level of
And-
previous.
Yep, and there's a
having
carnivorous
trouble hearing
sponge
me.
that's just up to the left of that hexactinellid sponge.
Come in
Kind
on
of
that second sponge.
Yeah, we can.
And if we
That would be
can
great.
make sure lasers are on as we pull out.
Copilot, can you check
Yes,
lasers?
I'll check lasers. Lasers are on.
Yep.
And I'm nearly above you.
Ship is in position.
Okay, I'm going to be, what, probably
2
3 minutes
amphipods
behind.
hanging on.
Yeah.
Video is clear.
All right, come on, that video.
Lasers captured. Thank you.
So, Copilot, I'm going to keep my
I think that
heading into
upper
the
one
current
might
and
be a
saddle.
cremation.
Okay.
Yep.
I'm right above
Oh,
you.
okay.
Backwards. Maybe I'm looking at it backwards and it's an amphipod.
Okay.
Should I look into the current?
You
At
should
least
look
somewhat?
in the direction of
we're
our
moving.
movement.
Great. I will continue to do that.
So, watch lead. I'm just keeping my head into the current because it helps with piloting. But
Roger
we are
that.
heading west.
Okay, thank you. Okay, so we are heading west.
Directly above you.
Do we
Along
want to
the
see this black coral? I'm assuming we have.
Any interest in going in further to get a closer look at the black coral?
That yellow thing below
Nope.
lasers.
Well, I was waiting for Tina to respond. Do you mean those white ones?
Yeah, the white ones.
Yeah, she does say she would like a close look if we can get to one.
Okay, so yeah, Josh, the next time you see one, if you can get closer to one,
copy.
please. So there's a fairly strong current here coming. Yep, go ahead.
No, nothing. I thought it was something really weird. I guess that's a crinoid that's on the rock to the left of the Metallogorgia. But so never mind. I thought it was something different.
Can see that feed on him better.
Yeah, come in, snap. Yeah,
Yep,
that.
sure is. Look at that, hanging onto the rock with its long cirri. And there's a little sponge over there that might actually,
Come in tight.
I don't know, demo sponge? I'm not sure.
Little top.
Right, I gotta move. Coming out slowly.
I think I'm actually stopped. But
That's alright. I'm gonna push out.
Ship stopped at 34. 3 minutes ago.
yeah, that seems right. And Nav, what's our off bottom time? Or how much time do we have left?
We had about an hour. Let me double check.
Okay, great.
Are you close enough to see that
Yeah,
center
1 hour and
player
1
up
minute. Off bottom
Okay,
time.
can we get a closer look at that white
3:38.
fan, please? Any of them?
Alright, I'll come into this one because it's in my heading.
Watch these, Nav.
Go ahead, Nav.
Just letting you know we have 1 hour left, and you would like us to continue west along this ridge, correct? Cool. Excellent. Thank you.
Okay, so we have an hour left in the dive. We're going to come and get a closer look at this.
Come on in, video.
Partial.
So which, is this one Stauropathes?
This one is the whitish one is the Bathopathes.
Bathopathes,
The
okay.
yellow one behind is the Stauropathes.
Okay.
This is Copilot.
Thank you. Go ahead.
I was going to say, I think Tina wanted to take a look at the base. Yeah, there we
take
go.
a look at the base. Yeah, there.
And I think I see a scale worm, polynode, polychaete in the main axis, and that's often where they're huddled.
I was just thinking this is a pretty good view of D2 from Sirius. I don't know if you want to capture or save it or anything like that. Don't want to get too much
They
closer
certainly
with our
are
heave.
whipping around in this current.
I'm thinking Tina wanted to see the base to see the polychaete, and we saw that actually further up.
Okay.
Probably a good view we've had.
Okay, thank you. Thanks, Josh.
You're welcome.
Pilots, looks like to me, but does 270 look like it'll stay on this ridge?
Yeah.
Okay.
I think so.
Look at this landscape, Patty.
It's a C-pin, but I believe we got it before.
Oh, we've gotten this before?
I think so.
Come on in, video.
Okay, is that one of the rock pens? It looks like the polyps are darker on this one.
Yeah, this is quite likely in the
Video
genus
is
Anthoptilum,
Sirius
so it's
HD4.
another C-pen. We've seen a few of them.
Yeah, I copy that.
This is
Yes,
what is
Sirius
now
is
referred to
4.
as rock pens, only really described 3 years ago, I believe, was the first publication. And it kind of
Rich
surprised the
rock
experts
diving
because
up to the right.
they're adapted to live in soft sediments with that inflated base. And so once we were able to get down into the deep sea with more ROVs and get good imagery, it became clear that at least some species in this group could suction themselves to hard substrates. So that was an interesting observation.
Josh, it's kind of like the claw, the manipulator that we were talking about yesterday, the soft claws.
Oh, yeah?
Yeah. The way that
Oh,
C-pen
yeah.
is attached to the bottom.
Exactly. Exactly.
Interesting coral at the bottom of the...
There's something very bushy down below
Pilot, do you
there,
want any
right at
lights
the
out?
bottom of the
They're all
screen.
stowed
I wonder if we
You
could
can
take a
leave them
quick look.
stowed right now.
Okay, fine.
Down
I
at
think
the-
they may have
Maybe another
wanted to leave them
Lillipathes,
stowed
Chris. Only the second
as
one
a
of the dive.
diagnostic test.
Is this the one that we're coming in on right now?
Okay, I think they had them out this morning.
Oh, they did.
Yeah, that's correct.
That's
lower
close.
Okay,
You
we'll
get
take
to
a
it.
look at that.
You can deploy the lower ones partially.
All right.
Come on in, video.
Okay, I'll do that.
Oh, wow.
I think I'm going to be eating my words in a second.
Wow, look at that.
I'm going to take your Titan.
What is that?
Wow. Haven't seen this before.
Look at that.
It's amazing.
Oh, okay. Well, at least, yeah, it's another black coral. So this may be like the one that we saw earlier in the dive. We'll see what Tina has to say about this. She's the expert in this group.
Not the easiest thing to image with that background.
Come out a little bit. Let's see if...
Oh, right behind the coral.
What about it?
See that little mound with the white around it?
Yes.
Why is it there? I know. There's one there.
Yeah, I know. As my daughter said, tell the lady who's whispering that we can hear her.
Chrysopapes. And you notice all
Look
these
at the
black
base
corals
of the
that we've
coral.
been giving
I'm showing
names
a
to
leak on the starboard
end
valve
with
pack.
pathies,
Oh, that's fine.
P-A-T-H-E-S.
Yeah,
Disregard
that's...
that.
Yeah.
The black corals belong to the order Antipatharia.
That, I think
Antipatharia
if you
translates
reset
as
the-
against disease.
The
Power
bell
cycle.
And the early
Slight
Greeks
rotate right.
believed that pulverizing the skeleton into a powder could be
Yeah, it's hard
used
to
for
rotate.
medicinal purposes, and therefore it was against disease. So that pathies part of it has just come to be a common usage in the genus names.
I had no idea. And here,
Isn't that
all those
amazing?
years I've been working on marine natural products, drug discovery, I had no idea. Okay, so you can, looks like you can see something.
Well, surely I didn't say it worked.
Well, I know, but we know these little, we're supposed to know these kind of myths,
All right, light
tales.
should be a little bit better.
I did not know that, although I'm sure my
Scott,